Welcome to Caring and Sharing, the Advanced Oncology Podcast. My name is Uta Schmidt-Straßburger, and I'm meeting Professor Alexander Kleger today. He is an internationally renowned physician-scientist who combines molecular oncology, stem cell biology, and their clinical translation in a unique way. I will point out three particularities why I think we should talk way more often. As the director and chair of the Institute of Molecular Oncology and Stem Cell Biology at Ulm University, he is leading a very recently developed field of research that is pursuing tumor modeling with organoids. Also, miniaturized organoids that help to make specific patient therapies more tangible. He has been appointed a Heisenberg professorship and directs the interdisciplinary division of Pancreatology, an intersection between the clinic and the lab with a clear focus. The results of research must reach the patients. And in 2023, he was awarded the German Cancer Prize in the experimental category. This is one of the highest achievements in German oncology. Currently, he receives funding by the German Research Foundation, DFG, via its highly competitive COSELEC program. Welcome to the podcast, dear Alex.
Uta, thanks a lot for the nice introduction. It's a pleasure to be here today.
As I just mentioned, you were awarded the German Cancer Prize for, among others, your work in modeling pancreatic ductal adenocarcinoma. In your opinion, what was the major contribution that qualified you for this prestigious award?
Oh my God. This is always a tough decision. I was not part of the reviewers committee, and I don't know what they really thought about, right? But so I believe that if you force me to do so I believe that the tipping was that we could demonstrate in recent years that pancreatic cancer, which is still one of the deadliest tumor diseases, can be described in a new way with stem cell-derived organoid models. Our approach intentionally went beyond the limits of classical tumor cell lines, or was actually tailored to go beyond these limits to get something new, a new model for a disease where we did not make that much progress in terms of research over the last decades. We could show that we can derive pancreatic cell types, either ductal cells or acinar cells, from human pluripotent stem cells. And these engineered cells can then be driven or pushed into early stages of tumor development by targeted genetic engineering. And this development subsequently permits us to observe tumor development in the dish, not only from a retrospective viewpoint as classical models do, but also in a kind of a time lapse. To model and get access under controlled conditions in the lab. And at the same time, we were able to gather more knowledge on the plasticity of the stem cell-derived ductal cells, and mainly the capability of these cells to tip into a pre-malignant or malignant program under certain genetic or also microenvironmental conditions, as we plan to do in the COSELEC program. I think this functional approach to model tumor biology was a central point why our work has received so much recognition at that time. And we could also underpin that in 2021 and 22 with a couple of high-ranked, high-impact papers to support this viewpoint.
Congratulations to that recognition, and also to the nice work that you've been doing, you and your team. So when entering your name into a search engine, one inevitably comes across the term organoids. You are using organoids not only as a model, but also as a functional system that mirrors the state of cells, their plasticity and responses to therapies. Could you just briefly describe for our listeners what organoids are and what the advantages of this model system are in comparison to classical cell lines or even mouse models?
So organoids are three-dimensional structures, and this is a strong contrast to cell culture models, which we have used over the last decades, which are usually 2D cultured on plastic. And these 3D structures comprise cells and matrix, so environment, to recapitulate a given environment resembling either the originally derived tissue or tumor in terms of function and morphology. These are the two major items which really makes a difference, 3D and matrix to model and take the environment into account. And I think that the particular strength of organoids is that they build a bridge between complexity and still control. Classical tumor cell lines, as I just mentioned, the 2D are genetically quite distant. From a primary tumor, there is a lot of clonal selection, and thereby, over the various cultures and years and decades of research, they became rather artificial. Still useful, but artificial to a certain degree. On the other hand, we have mouse models, and here we have the complexity of an organ, a living organism. But as we all know, it's a mouse and not a human. So the mouse does not fully recapitulate a human being. We also know that certain genes have a completely different context in the mouse than in humans. And thereby, there are many advantages of the organoid systems as compared to the mouse, but also disadvantages because we lack certain components of the environment on the other hand. Still, there are a lot of strengths. Organoids, particularly the ones that we are creating from human pluripotent stem cells or directly from patient material, offer an entirely different approach. They maintain the characteristics of the tissue and of the tumor. I told you architecture, the degree of differentiation, molecular signatures. And at the same time, we can manipulate them. We can introduce defined single mutations or various mutations using CRISPR-Cas technologies, for example. We can manipulate thereby signal transduction pathways, or even more exciting, we can eventually also test responses to therapy. And this is possible in a system that can be constructed around or from a patient. And therefore, for me, organoids are not a replacement model for mice, but a new principle, a complementary principle that functionally and dynamically permits studying tumor biology in the lab as we were looking through a pinhole into a patient. And this is not possible in mice.
Thank you, Alex, for very nicely explaining to us the direction you and your team are pursuing with organoids grown of stem cells. But as you just pointed out, organoids can also be isolated directly from tumor tissue. Could you elaborate a little bit more how that works and where the differences to the stem cell-based models are?
Absolutely. This is, from my point of view, an as exciting field as the stem cell models, right? Apart from the stem cell derived models, which we really have to differentiate over time, over many weeks, to the respective cell type, we are also working with directly patient-derived organoids. So we call them PDOs. This is the abbreviation which I will use throughout the further interview. And these PDOs are isolated directly from tumor tissue, either from a biopsy or a surgical resection. And this procedure allows us to create these individual mini tumors within a few weeks. And these models maintain many properties of the original tumor. And when we started with all this research, we asked from the very beginning the question, not only in terms of to what extent do they molecular recap a given tumor, but to what extent can we directly make use of them to eventually predict chemotherapy response. So we use them to perform so-called pharmacotyping. So we test, given a set of drugs on the organoids resembling the tumors, to eventually predict whether these organoids can say this patient would respond to this therapy or not. I found this super interesting that we could see the predictive value of these PDOs in this pilot study, particularly in treatment-naive patients. This shows that this system is not only specifically relevant, but could also offer a real clinical benefit, for instance, in first-line therapies, but also in subsequent lines. But what I would like to emphasize at this point, we need clinical trials. All this, what I just told you, are retrospective correlations, lab studies, all experimental. So we need the clinical trials, which we're also pursuing here in Ulm, to really test and probe and challenge the PDO value for their predictive capacity to guide clinical decision-making in patients.
Thank you. The results you've been just describing were generated in 2021 and before. So I wonder what happened since then. It's 4 years, and I think you've been quite busy, as always.
Many things happened, and I think particularly great research hopefully happened. But this was also a time when we realized the eventual value of pharmacotyping in the use case pancreatic cancer. We had the idea to bring organoid medicine also to the broader community here at Ulm University, and thereby somehow seed organoid research to other labs here on campus, setting stage for research consortia and proposals. And I asked at that time the Dean of Faculty whether it would be a cool idea to launch a core facility for organoids as a kind of a central service unit to develop patient-specific organoid cultures, assist other labs in deriving the tissue, and use these organoids eventually in clinically relevant decision-making in the future. And since its inauguration, this core facility service has further evolved and does now offer a wide spectrum of methods, such as the establishment, cultivation of tumor organoids. We have thereby a super large biobank. We had almost 300 fully characterized organoids now in our freezers with pharmacotyping profiles. Pharmacological screening becomes possible. We offer scientific services such as project planning, project assistance. We assist for revision of papers with a heavy workload in terms of organoid medicine, and so on and so forth. We have also welcomed researchers from nationally, but also internationally, to use our SOPs for their work in their labs. And we are really proud what we could develop over the last years in the organoid core facility, particularly also with the staff. We have a brilliant technical head at the core facility, Dr. Jessica Lindenmayer. She's also actively involved in research projects. And she's the driving force of the organoid core facility in Ulm. A major progress for the integration was also the integration of cutting-edge technologies, such as our 3D bioprinter. We also have a small pipetting robot, and these devices help to accelerate and standardize the generation of a tumor environment and of an organoid culture, and they definitely help to improve the reproducibility of our results. But we do not only isolate organoids of pancreatic cancer, so this was really important for us. We really wanted to widen the spectrum. Now we are creating also organoid cultures for mammary carcinoma, urothelial carcinoma, and several other entities which we have now in our living biobank. We also published now from the various papers just recently for bladder cancer with the support of the organoid core facility, and thereby it really happened that we could seed organoid medicine to various research institutes and hospitals on campus. Like this, we can enlarge the applicability of the models and develop also novel therapeutic approaches for a bigger patient clientele at Ulm University. In summary, I would say the core facility organoid, as one of the first of its kind in Germany, has evolved into an important platform for translationally oriented cancer research that advances both basic research as well as clinical application.
You get so excited talking about all this work. So why is this topic so dear to you?
So maybe I just, from the very beginning, I always liked research, but I also like medicine. So I'm particularly touched by everything that we do because of the proximity to the patients, despite all the high tech around that we are using. And I do not mean the complex cell models or molecular mechanisms, but the hope to be able to really to make a contribution to patient care, to make therapeutic decision-making faster, to better understand tumors, and they really make a contribution to the cancer research field. And this is, I think, particularly important when we talk about pancreatic cancer, when time is really pressing and the therapeutic options are super limited usually. And thereby a functional model like patient-derived organoids or stem cell-derived organoids for the early stages can really eventually really change something. Eventually, I don't know, but eventually. And it is not just about data points, but really whether a therapy works or not, and whether we can understand a tumor or not. And I'm touched how so many people, multidisciplinary, from the hospitals, labs, ITs, bioinformatics, are working together to bring these systems into patient care. And this is a form of modern medicine that is not only challenging on a technical, but also on a human level. And this makes it, for me, somehow really relevant. And that's the reason why I still like my job. I always tell my kids, I'm happy to go to work, and this is the aim why you go to school.
I'm with you there 100%. Thank you so much, dear Alex, for taking the time and joining us today. It was a true highlight to get a glimpse into the world of organoids and personalized cancer medicine. I wish you and your team loads of success, and I'm looking forward to meeting you again in person soon.
Thanks, Uta. It was great. Great to meet you today.